Good day, this is Professor Will, CBMD, PhD, and today's Wednesday, October 20th, 2021. It is 12:09 and 32 seconds, a.m. Eastern Standard Time. And today's research, DNA robots. So let's hop to it here. How to program DNA robots to poke and prod cell membranes. Date October 15th, 2021. Source, University of New South Wales. A discovery how to build little blocks of DNA and get them to stick to lipids has implications for biosensing and mRNA vaccines. It discovered the best way to design and build DNA nanostructures to effectively manipulate synthetic liposomes, tiny bubbles which have traditionally been used to deliver drugs for cancer and other diseases. But by modifying the shape, porosity, and reactivity of liposomes, there are far greater applications, such as building small molecular systems that sense their environment and respond to a signal to release a cargo, such as a drug molecule when it nears its target. Researchers discovered how to build little blocks out of DNA and worked out how best to label these blocks with cholesterol to get them to stick to lipids, the main constituents of plant and animal cells. One major application of the study is biosensing. You can stick some droplets in a person or patient. As it moves through the body, it records local environment, processes this and delivers a result so you can read out the local environment. Liposome nanotechnology has shot into prominence with the use of liposome alongside RNA vaccines, such as the Pfizer and Moderna COVID-19 vaccines. And this work shows new ways to coral liposomes into place and then pop them open at just the right time. What is better is because they are built from the bottom up, out of individual parts they design. They can easily bolt in and out different components to change the way they work. Previously, scientists struggled to find the right buffer conditions for lipids and liposomes to make sure that their DNA computers actually stuck to the liposomes. Researchers struggled with the best way to decorate the DNA with cholesterols so that it would not only go to the membrane, but stay there as long as was needed. Is it better at the edge, the center, heaps of them, few of them, close as possible to structure, or as far as possible? Researchers looked at all these things and showed that they could make good conditions for DNA structures to bind to liposomes reliably and do something. Researchers say membranes are critical in life as they allow compartments to form, and therefore different types of tissue and cells to be separated. And this all relies on membranes being generally quite impermeable. Here they have built today new DNA technology where they can punch holes in membranes on demand to be able to pass important signals across a membrane. And this is ultimately the basis in life of how cells communicate with each other, and how something useful can be made in one cell and then exported to be used elsewhere. Alternately, in pathogens, membranes can be disrupted to destroy cells, or viruses can sneak into cells to replicate themselves. The scientists will next work on how to control DNA-based pores that can be triggered with light to develop synthetic retinas out of entirely novel parts. Good day, this is Professor Will, CBMD, PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.